I want to talk a little bit about mating biology. This isn't really my work as much as it is the work of Gudrun Koeniger and her husband Nicholas. Both are PhDs from Germany. They have worked with the Rutners in Germany, and when the Rutners stepped away from the big institute there in Oberursel, Nicholas was the director for a number of years. Gudrun stayed home, raised the kids, did hard science, edited journals. One of the things they've looked at, and of course something I'm fascinated in, is the whole idea of mating biology, mating behavior of bees, and why is it so strange? Why do bees do things the way they do it, and what's going on? So why do bees have drone congregation areas? Well, the answer is it helps to prevent inbreeding. The queens fly quite a distance while the drones don't. And this prevents inbreeding so that a queen is not going to mate with her brothers and preventing an incestuous relationship. Inbreeding does a number of things to a beehive. One of the things we normally associate with is spotty brood. Now, I'm not sure this is actually the result of inbreeding, because sometimes spotty brood is a result of mites and other diseases. I think this is one of the diseases we're having trouble with. But spotty brood from inbreeding appears when you have the same sex allele present in an individual bee. And the bees have an interesting technique for taking care of that problem. When you have the same sex allele, we call those individuals diploid drones. When the larvae hatch, the worker bees eat them. It's a direct and immediate way, thank you, a direct and immediate way of taking care of a problem. Polish researchers have reared out these diploid drones in the laboratory in incubators, feeding them royal jelly and then drone food, and found that they're nothing special. They don't bring any superpowers to the beehive. There's no cape on their back. As a matter of fact, they're pretty disappointing when it comes to their genetic potential. As a beekeeper, you want to avoid inbreeding because inbred colonies are bigger. The more bees you have, the more things those bees can do, like make honey, collect pollen, pollinate crops, and sting you. Well, I thought I'd throw that in, just you might be interested. So inbred colonies at the same time tend to be smaller, less thrifty, less efficient, and they're more likely to have a disease or three, or mites. Now, to get into this whole business of the haploid, diploid, we'll use this graphic. So an unfertilized egg, if you've taken your Bee Biology 101, read Dewey Caron's book, my book on the field guide, you'll know that if a single chromosome is present, that's the drone. It's haploid. So that's real easy. Queen goes to a cell. She measures the cell, we think. She deposits an egg, and she doesn't release any sperm. Unfertilized, that individual becomes the haploid individual that we happily call the drone. Females, though, are diploid. They have two sets of chromosomes. And the ones on your right, 1, 2, 3, 4, 5 of them, have different colors representing different alleles. But the one there sort of in the center has the same red. For those of you who can't see red, that's red. So it is an indicator that the two alleles for sex determination are identical, and it becomes, instead of becoming a female, it becomes a drone. But a diploid drone, which are the ones the bees are going to eat. So it's a complicated story. It's one of those things that, how did that happen? The other thing that I ask myself is, how did this isolated drone congregation behavior start? Because when you go to something like bumblebees and you look at their sex life, you say, well, this is boring. They have no shame whatsoever. They'll copulate on a laboratory bench. If you put a male and a female together, say, hey, let's go. And so here's, those are the same species. You see the difference, the males on the bottom, females on the top of the slide anyway. And notice all the mites on the female. And bumblebees, these aren't the mites that we have in the beehive. These are different species specific to bumblebees, and some of them have an enormous problem with mites. I've seen bumblebee nests that are just slimy with, a special movie effect kind of thing, with all the mites crawling all over the nest, the bees, the food, everything. So that's a sidebar. But why do bumblebees not have this isolation in mating? Or more exactly, why do honeybees isolate themselves with a DCA, a drone congregation area? And where is a drone congregation area? Well, my friend Carl out in England, he wanted to find out where they are, and he went out looking, and he was walking in the neighborhood, didn't find one, went back to his apiary. The balloon went up with a little pheromone at the bottom of the balloon, and bang, he could hear the roar of the drones. So his apiary happened to be a location of a drone congregation area. We still don't fully understand what makes a spot a DCA. We do know that they are permanent. They go on from year to year to year, and yet no one's found the graffiti inside the beehive where the bees have chiseled out, the equivalent, the biological equivalent of for a good time. You'll find the boys out at the center of this hayfield, whatever it may be. So here's an area, you have a flight area. It's going to be up in the sky, 50 to 200 feet. There's some indication now that the different races of honeybees stratify, like going up an elevator. First floor, Apis mellifera mellifera. Second floor would be Apis mellifera ligustica. And all the way up at the top, there might be some place for Apis cerana. I'm just using that as a joking example. We don't know where these are, but there's some indication. And Debbie Delaney in Delaware has looked at some of this. We can go out. Where's my EAS volunteer? Who's going to do it? Right there. So you're going to set up, going to get some weather balloons. You're going to get a party balloons. Have a party, get the helium. Don't use it all on funny voice effects. And a cage which has some queen pheromone. We have a little bottle of Everclear that we keep, and we just drop any queen that's done her job, whether it's an old virgin queen, which just seems sad, but she won't mate right, or an old queen. We just drop them in there. And the alcohol that's in there, once it's evaporated, is going to provide the residue of a queen pheromone. So I don't think you need to buy it. I think you can probably find it. The other thing which is really, really strange is that the ratio of drones to queens or queens to drones in a DCA is really, really skewed toward lots of drones and not very many females. It's about 2,000 drones for one queen. That's not a very good statistic for the drone, is it? And so in a thriving colony, we estimate that they might produce between 5 and 25,000 drones over the course of a season, depending on the stock, the year, the strength of the hive, all those obvious things. Whereas a normal colony was maybe a swarm or two, maybe a supersedure, on average they produce about 6. So we might actually overstate that. There might be fewer queens and more drones than 2,000 to 1. Now, let's do a little hard thinking. And I like to think that bees are bankers. They have a strong accounting department. And in the accounting department, they say, look, if we send that queen out, if we send out 500 drones, that might be the number of drones that are flying that day for a particular colony. If we send them out a mile to mate, a mile, okay? That's 500 drones that are going out a mile. There's a problem. Drones only last about 25 minutes in the air, and they have to come back and refuel. That's included in the 25 minutes. So if a drone goes out for 10 minutes, takes to cover that mile, he's only got 5 minutes of patrol time before he has to turn around and come back to the hive and get some more honey. Now, the drone may go out 3 to 5 times, 6 times a day on a good day. It may get cut off by the weather, whatever else. But it's not very efficient because those drones are going to consume a lot of honey. But if the queen is the one that travels the distance, so the accounting department went to our primordial queen and said, it's your job to fly the long distance. And the queen's argument is, wait a minute, I'm a sweet young queen. You want me to go out there and fly a mile someplace I've never been, meet a bunch of drones I've never seen before. They may be some other race, you know. And I have to fight off the birds, the dragonflies, run the risk of being caught in a thunderstorm. And I don't remember what hive I came on. The damn beekeeper paints all these hives white. All right. So it's a high-risk proposition for the queen. But that's what they do. The queen's going out and covering the geography. So our drones are going to fly an average of 25 minutes per flight. They're making 3 to 6 flights per day. Local flight means they have less time to get to the DCA, so they have more time in the DCA. I don't know if I said that right. So it's to the colony's advantage for the drones to stay local. And the queen flies some distance, on average, a mile away. Now, most new beekeepers say, I thought my queens and my drones were going to mate with each other. Nope. You don't want that. It's going to result in inbreeding. So you want to have these queens of yours to go out and mate with your neighbors' males. So now all of a sudden you're really interested in what does your neighbor beekeeper have in her beehive. What kind of bees are you keeping, Catherine? What do you got in there? Have you got acceptable drones for my lovely virgin queens? And of course, this is a conversation beekeepers have been having, and we talk about it when we say we want to set up congregation areas. You can go out, set up find a DCA, and then lower the balloon so that the drones come down with the pheromone, and then you can sample those drones. The more modern technique is take your balloon, and there's a cage here that has an opening at the bottom, and the pheromone is inside that cage. Wow. So the drones fly up, and they seem to be stuck and forward. They can't reverse out. So they're in there, and you as a beekeeper can sample the drones that are in that area. Pretty clever, huh? Now, remember, you've only got 25 minutes maximum to do anything with those drones, so you better work quickly. So here's the drones are in this trap. They're attracted to queen pheromone. And now our leaders in this, the Rootners, the Koenigers, in this country, we've had people like Norm Gary from the University of California when he was a grad student at Cornell. And other people have gone out and marked these. The most recent group of people in the U.S. that studied this were Chip Taylor and Jerry Loper, Chip from Kansas, Jerry Loper from the USDA in Tucson, And they used a little radar device. And people always ask me, well, have they put little reflectors and transponders and you guys with all your high-tech toys? Are we inside the Beltway? Yes. So you guys know more about this stuff than I do. And I'm not sure I want to hear it. I want a drone that chases after the drones in the DCA. You know, one that's got a joystick. That's my ambition. Chase after them and photograph this stuff without having them tethered. So marking, we'll go back. If you are in a hive, you can mark all the drones in a particular hive on their thorax. So all the reds are from one hive. And then when you collect drones in these cages, you put paint on their abdomens. So each is a different color. And then you can sit down and analyze all these data and come up with some conclusions on what's going on in this particular situation. This is important because I'm going to spend a lot of time talking about it. So here's the results of different apiaries, not hives, but apiaries. The number of drones in each apiary. And the one on your left is near, the one on the right is distant. And you can see there's some variation, but the distance, you know, there's some that they go go far away, but there's some like T1, very few drones go anywhere. Or maybe they're the lazy ones, I don't know. So relatively few drones are going some distance away to that to faraway DCAs. I'll spit it out. So queens fly low, and they average a mile. And this reduces the chance that she's going to mate with a brother and all of those horrible problems that you're going to have with inbreeding, incest. It's to the colony's advantage that the drones stay close. If we have, in the example, I have 500 drones, they stay close to the hive rather than flying a mile because they consume fewer stores. So you have to decide which is going to get stuck with the distance flying. And I said that the primordial accountants in the beehive said, girl, you got to do the heavy work. Never heard that before. Right? The men get to stay close, go back, eat a lot. The queen, in theory, she's going to fly out 10 minutes. She's got 5 minutes in the DCA and then fly home. So what is the other adaptation that the bees have gone to? Really fast sex. That the coupling of a female to a male lasts a giant 2 seconds. So another drone can mount that queen and she can be fully mated in 5 minutes. No problem. So by swapping the drone line and the queen line over 3 years, the Rutners or the Koenigers were able to look at this phenomenon. And what they found was that it wasn't the location that determined who flies the distance. It was always the gender of the bee that determined who's flying in that distance and who's not. Also, by looking at the drones on a purely statistical basis, based on a distance from the hive, you can see there's a nice correlation there that drones are staying closer to the hive within 1,000 meters. And few of them are going, there's one there, almost 4,000 meters. What was his story? You know, well, what I like to say at this point is, isn't Mother Nature wonderful? She allows for all possibilities. So, is there a chance that a particular queen would mate with her brother? Absolutely. But what she's doing is reducing the probability, the chance that she's going to mate with multiple brothers. And that becomes important here in a couple of minutes. Now, putting any queen I'm just going to make a blanket statement. Any queen from any hive runs a 25% of not mating. 20% of the time, she's not going to make it back. She's going to be a dragonfly lunch, bird snack number 3, whatever it is you want to chalk that up to. And that 75% success rate, 25% success rate is based on 2 observations. 1, 30 years of data from mating stations in northern Germany. And number 2, the fact that I always feel like that I'm losing about a quarter of my mating, no matter what hive it is, whether it's a mating nuke, a big, strong, booming colony that they send out queens and they don't make it back, secondary swarms, after swarms. Those queens don't mate until after they get in the new hive. And many beekeepers say, well, I took apart all the bees and they're counted, all the bees in my after swarm, and there were 6,000 worker bees, 403 drones, and 8 queens, all small virgins. They haven't swollen up yet. So the crossing, excuse me, the line for mating is after the colony swarms. That means, think about it. That colony has, the parent colony has invested all that energy into swarming. And the after swarms that are generated only have a 75% chance of the queen getting mated, much less being established. You know, and Tom Seeley, in his first book, Honeybee Ecology, he says that in nature, natural hives have a 1 in 6 chance, 1 in 6 of those colonies that swarm will make it to their first birthday. 5 out of 6 don't make it. So that's where I say beekeepers are a big help to beekeeping if the beekeeper is able to really help their bees by what? Feeding them, disease management, wintering, whatever else needs to be done. Now, when a queen comes back from her mating flight, she has that mating sign in her sting chamber. In the mating process, each successive drone pulls that out with a hairy patch on his body. But the last queen comes back with the last drone's mating sign, and the worker bees have to remove it. Now, what's the purpose of this? Some people feel it helps keep the sperm that are in the body of the queen from leaking out, and we'll show you that in a minute. Sperm, well, you know, we're not talking about one spermatozoa. We're talking about millions. And so the bees have evolved the existence of a spermatheca, a sperm storage structure. It's a small, clear sphere in the tip of the bees, the work of the queen bee's abdomen, that's covered with a very fuzzy-looking substance, which is actually the tracheal material that go to the breathing tubes in the abdomen. You know, each segment has its own breathing tube in the spiracles. So these that you see in the upper right then, where it says it's a TR, tracheal tubing, that's providing oxygen. Now, the spermatheca also is floating. It's positioned. It's attached to the reproductive structure, but this whole sphere, except for one little tube, is floating in the hemolymph of the queen bee, the blood. So we assume that the hemolymph is entering and providing nutrition for the sperm that are stored there. Thirdly, you see a kind of an M-shaped DR on this. That is a gland that is providing, we're not really sure, but when Gudrun Kronecker very carefully dissected queens and removed part or all of that spermathecal gland, the sperm died. Queen lived, sperm died. So that tells me, and it tells them, that it's producing something, and there's now some biochemistry on this that's indicating it's pretty fancy stuff. So you need to have that. So the spermatheca, at first glance, looks like it's a really simple structure, but it's not. It's a very complicated structure. The other thing is it holds a lot of sperm. So if you freeze the spermatheca and use the microtome, you can get something that looks like that. So you've got the trachea around TR, this SP or the sperm, and the W is the wall of the spermatheca. How many sperm? Between 5 and 8 million in a well-mated queen. Commercially, somebody looked at the number of sperm in commercial queens, and it's between 3.5 and 5 million, which is kind of close. So we're all right. So a queen is producing pheromones. We see that by the bees that are attracted to a queen. A queen starts producing pheromones before she emerges from a queen cell. So there's an odor there. There's probably a unique odor produced by a queen as soon as the bees have given her that responsibility. So an emergency queen cell, you go in and step on the queen, and the bees replace or build half a dozen to maybe 100 replacement queen cells. They all are clearly identified to the worker bees as being queen cells. Now, many colonies will trim the number of queen cells they've got. Big debate in this whole argument about superfamilies. Another topic to find somebody to talk about. What is a superfamily? Well, basically you've got queen bees that are mated with multiple drones. So the worker bees that share the same drone are called super sisters, rather than the other drones that are just sisters. And their degree of relatedness here is changed. Well, there's been a lot of work to say, okay, these bees that are super sisters, they're going to pick out their sister queen candidate. And yet, that hasn't been proven. Because it looks like the bees toe a fine line about, rather than favoring one queen that might be particularly, maybe something defective about it, maybe a minute too old. And the bees always go with the individual they feel is the best for the colony. The queens, of course, they're attracted to each other, not in a good way. Sister queens. Now, mothers and daughters don't do this, unlike humans. No fighting here. But sisters, like humans, they fight. So, and the queen will try to sting her rival in the wing pit, in the membrane, the very underneath the wings. Does the amount of pheromone in a hive vary? Yes, it does. Young queens, so you have a small amount of pheromone that's being produced in a queen cell that increases in a new virgin queen, newly emerged virgin queen, in the first 12 hours. And then by the time that virgin queen mates and starts to lay, her volume and complexity of chemicals increases. But where does she hit peak pheromone production? About a year later. That's when she's in her prime. And then she starts to slowly lose some of this pheromone production, which I think may, in many cases, be correlated with a slight reduction in brood rearing until you get to supersedure. And that's when the bees, quite often, you see a big drop in the brood area. If everybody else has got 8 frames of brood, a colony that's about to supersede may be down to 3 to 5 frames of brood. So this seems to be one of the triggers that's involved in that supersedure process. All right, so here's a drone mounting a queen in a DCA. The queen is tethered. She's on camera, on a pole, rotating around. And several people have done this now. I think Gudrun was the first person to do it, about 1969 or so. And giving us photos like this so that we can see the one drone has mated, is copulated. The problem is the queen is tethered. She's tied, and she does not respond completely normally. So that's where we have to kind of fill in the pieces. A second drone will come along and knock the drone off, pull him off, so that the second drone is able to mount the queen. Well, people say, well, how does a queen know when it's enough? Is it a spirit of the moment? And we don't have a good answer. The Kernigers think it's the number of times she opens the sperm chamber and allows a drone to let his endophallus go into the queen. You know, when I was a graduate student, we thought that queens mated 5 or 6 times. This is going back a few years. And then for many years we thought it was around 8 or 10. And most recently we're now up to an average of 15 drones per queen. But the exceptional queen, based on DNA analysis of the different sperm in the spermatheca, we now have got one individual queen that mated with 60 drones.
Probably not in one flight.
Oh, how would we know? So that just indicates that there's a lot of variation in the numbers. So I like to say that we have to be prepared as beekeepers, make sure we have drone production for 15 to 20 sexual partners for each queen. And I always play devil's advocate. For every drone that's successful in mating, how many do you have to put into the air? 10? 100? 1,000? No one has a really good handle on that kind of thing. Once the drone mates, the sperm are held in by the velfold, which is some fleshy tissue that prevents the sperm from leaking out of the flying queen. It allows the queen to mate repetitively. So here you see the 2 ovaries, and the 2 lateral oviducts are filled with sperm. How many? As many as 80 to 120 million sperm in that queen. If you do instrumental insemination, we estimated we were putting in somewhere between 50 and 75,000 sperm in 8 microliters. So we're providing, a low, estimate of what's going on here. But here's the kicker. Only 5 to 8 million sperm end up in the spermatheca, unless you're a commercial beekeeper, and it's even lower than that. So those sperm have to migrate in. Just think, the drone had to compete to mate. The sperm have to compete to migrate into the spermatheca, into a little spermathecal duct. And then when the sperm are released during fertilization, they have to compete to fertilize the egg. So that's a complicated biology. If we look at a colony, and we just ranked them here by the number of offspring of 13 different drones. And you can see the percentage. So D1 is a drone. D13 is a drone. But they are not represented equally in the hive. Everyone understand what this little graph shows? Shows you have multiple fathers, but they're not equal. The amount of sperm in the spermatheca at this moment being expressed by the workers. If you analyze the worker's DNA, looks like this. Now this can change. And not all drones contain or pass on all behaviors. So here are the drones that contribute to guard bee behavior. We can go back. Those are the same drones. But, you know, not all of them have that trait. And we look at another characteristic. This is for water collection, that even fewer carry that trait. So what's this starting to imply? What's this going to wake up your brain cells? Hey, there's a benefit for having all these drones. Because if a queen only mates with one drone, there's a good chance nobody's going to gather any water or guard the hive. So this provides greater diversity within the colony. Looking at it from a different perspective, Dave Tarpey and Tom Seeley and others have looked at multiple drone inseminations versus single drone inseminations. They've shown a direct correlation between a colony's ability to withstand diseases by an increased number of drones that are used in the insemination process. So the more drones, the more likely that colony is going to be able to withstand diseases, mites. You get these other characteristics, these other behaviors involved. One I like is that the multiple drones are responsible for a colony that's better able to regulate its hive temperature. Single drone inseminations can't do that as well. And I remember when I kept bees in Florida with the Daydamp program, that we had colonies that we constantly had to add brood from other colonies, so that they would stay alive. And to quote my good friend, the late Steve Tabor, all an inbred queen wants to do is die. So we want to talk about drone development as well. And here's something we all need to be aware of. Drones really aren't designed to be highly fertile on less than good nutrition conditions. You say, what? The first challenge that a colony has is to produce drones in the drone season, which starts here, what, in March? Your drones are appearing in March? And what's in bloom in March? Maple, if you're lucky. What else? But those drone larvae have to be fed pollen. They need to have a good nutrition for them to make sperm cells. So here's your question for your master beekeeper exam. When are sperm created in the drone larvae? When are they formed? So now the larvae go into pupal stage, they stop making sperm. That the physiology of the drone at that point has to be to go from this primitive larval existence to the very complicated organism that we call a drone. So, in the adult drone, a well-produced drone has testes that are loaded with sperm. And then, during the first couple of days of feeding, after the drone emerges, that drone has to have more pollen, more protein, for that sperm to migrate from the testes to the seminal vesicles. And you say, oh, why do they have to worry about that? Because the sperm has to be in the seminal vesicles, so it's present during that 2-second sex. Because that's not going to allow any time for those sperm to migrate during 2 seconds. So they've got to be there. It's got to be ready. So there's 2 times, 2 episodes when these drones have to be ready, or well fed. Now, Some of you buy queens from elsewhere. And some of you want queens as early as you can get them, right? But you should know that the drones that mated with those queens, they had to start production before the queen breeders started producing the queens. You take my class on queen rearing, I teach you that the timing date is when the drones have purple eyes. That's when you can graft for queen rearing. So those drones take longer. It takes them longer to develop, 24 days rather than 16. And they're slower to reach sexual maturity than queens. My friends at Kroeninger say, look, it's like Mother Nature has put all of the tricks she has in the queen, and the drone just keeps plodding along because the colony can make lots of them. The problem is Mother Nature didn't know that we'd have beekeepers in the South or in California or Hawaii trying to produce tens of thousands of queens early in the year. That one cold snap may come in and destroy the pollen that's going to feed a week's worth of drones. And I can't tell you how many days it's going to take to destroy the sperm. But it's significant when you get a really cold weather pattern, when they cut down and so forth. Well, I'm beating that one up pretty bad. Now, the other thing is, how long does it take that drone to reach sexual prime? So this is the number of days after emergence and the number of sperm. You got around 10 million sperm potential in each drone. So look, one drone can take care of a whole queen, right? That's not what we're doing. But we don't have any sperm migration till a little bit on the third day, but it isn't until the 12th and 13th day that you reach your maximum sperm in the seminal vesicles. Oh boy. That means those drones take longer to develop. Yes, you've got mating on the fourth day, potentially. But the queen that gets mated with all 4-day-old drones, she's not going to get the amount of sperm that if she waits and mates with all 15-day-old drones. Again, our passion as beekeepers to advance the season causes problems. A lot of queen breeders say, well, I went out and I saw all these drones flying. I got a lot of drone flight. Hundreds of drones. Yeah, they're taking cleansing flights. Orientation flights. And so it wasn't until at the 8th day that we start seeing some mating flights, and then they're short. It isn't until we're up around 20 days, 18, 20 days, that we maximum out our drones going out for that full 25 minutes. Wow. It's like these drones start out with training wheels. They have to learn. Is there teaching going on? I don't want to get in there. All right. Now here's the kicker. It takes about 10,000 drones to maintain a DCA. If a DCA doesn't have enough drones in it, the bees close it down. How and why and all that stuff I can't tell you. But because of the time it takes drones to fly back and forth, that means you need another 5,000 drones in transit. So every DCA is 15,000 drones. All right, do the math. How many colonies do your bees produce a year? How many drones do your colonies produce a year? How many colonies does it take to put 15,000 drones out there? What'd you say? What percentage of drones? Depends on the colony. The statistics I've seen is that one colony may have as few as 5,000 drones a year, or as high as around 20,000 drones in a year. Big difference. So this variation here is going to be another variable, you know, that we have to keep in mind for all that. Well, Bad Larry, he didn't find this book, but we do have a new book on this subject that talks about it. And it was a collaborative project between the Koenigers, Jamie Ellis, and myself to get it in print.